Okay, our next speaker is Maria Agustina Rodriguez, and her talk is titled Lacticaseibacillus paracasei B2, isolated from water kefir, exhibits probiotic potential for use in honeybee feeding.
The use of beneficial microorganisms is a sustainable strategy to improve colony health and control honeybee disease. Probiotics are defined as live microorganisms that, when administered in adequate amounts, confer a health benefit on the host. This is the definition of the International Scientific Association for Probiotics and Prebiotics, ISAP. In previous studies, we have isolated the lactic acid bacteria Lacticaseibacillus paracasei B2 from water kefir and evaluated it through several tests in our laboratory. We found that the bacteria had antimicrobial activity in vitro against the honeybee pathogens Bacillus larvae and Scopher aureus. Moreover, when incorporated into the diet, the bacterial strain was non-toxic to bee larvae. And also the strain was able to tolerate stress conditions such as acidity, osmolarity, and temperature variations, highlighting its probiotic potential. Here we can see a picture of the bacteria growing on a petri dish with the man rugosa and sharp culture medium. In the second picture, we can see the water kefir, that is a fermented beverage from which we have isolated the bacteria. Then we can see two petri dishes where we can see the antimicrobial activity of the bacteria against bee larvae and the fungus Ascosphaerapis. And finally, a bee larvae reared in the laboratory and fed with the bacterial strain. In this study, the aim of the research was to determine the effect of probiotic supplementation on the nutritional condition of honeybees and to evaluate the ability of the bacterial strain to survive under field conditions in hives. To conduct this study, we first formulated the supplementary products in the laboratory. The 4 supplements that we prepared were the control group that comprised the standard 170 grams patty without additives. Here we can see a picture of the final product. The vehicle group comprised the standard product with 35 milliliters of sterile man rugosa and shelf culture medium. The supernatant group comprised the standard product with 35 milliliters of cell-free supernatant of the potential probiotic strain. And the probiotic group comprised the standard product with 35 milliliters of growth culture of the potential probiotic strain. The experimental design consisted of a field essay conducted at the experimental apiary of the agronomy department at Universidad Nacional del Sur during the month of October. For that, 10 productive Langstroth honeybee hives were selected. 7 colonies received a total of 4 supplementations, and the 3 remaining hives were used as natural controls and did not receive any supplementation. In order to assess the preference of the bees for the nutritional products, the 4 supplements were placed over the brood chamber frames in each colony during 7 days. As we can see in the picture, each day we retired the product and we put the new product over the frames. And every week we changed the place where we located the supplement. For example, if the first week the control group was here, then the following week was next in the next place, then here, and finally here. So after 1 week of supplementation, consumption was calculated as a difference between initial and remaining product weight and expressed in grams per colony. To evaluate the nutritional condition of the honeybees, nurse bee samples were collected from the brood frames into plastic receptacles on the first and the last day of the essay after 1 month. and the analysis of fat body reserves was performed in the laboratory. Finally, we studied the survival of the bacteria in the supplement. For that, we compared the number of the colony-forming units of the strain in the supplement before hive exposure and after 7 days using the drop plate method. The results obtained in this study regarding the preference assay, Group 2, that is the vehicle treatment with the mangrogosa shelf culture medium, was the least consumed by honeybees, while the probiotic supplements, Group 3 with the cell-free supernatant and Group 4 with the broth culture, were not significantly different from the control group without additives. When analyzing the nutritional condition of honeybees, after 1 month, these fed oil supplements showed a significant increase in fat body reserves, and a similar increase was also observed in these from natural control hives. In terms of the survival, the strain remains viable in the supplement for up to 1 week in the hives, although a reduction in the colony-forming unit counts was observed from 6.76 logarithm to 4.69 logarithm. So the modified supplements containing the bacterial probiotic strain delivered as cell-free supernatant and broth culture were well accepted and consumed by honeybees. After 1 month, these fed oil supplements showed a significant increase in fatty body reserves. However, this increment was also observed in bees from natural control hives, that is, hives that did not receive any supplementation. This may be attributed to variations in the inflow of pollen sources within the hive. Finally, due to the number of microorganisms to be used as a probiotic is crucial to exert the desired health benefits on the host, future formulations should aim for higher initial dose. The conclusions of this study were supplementation with either cell-free supernatant or live cultures appears promising for improving nutritional status of Apis mellifera. The results reported in this study are encouraging and highlight the role of Lacticaseibacillus paracasei B2 as a probiotic for honeybees. And now I would like to talk a bit about a new field essay that we are performing that consists of feeding honeybees with a supplement product, just the same that I present in this study, and also with sugar syrup. But in this case, we want to incorporate the potential probiotic yeast strain, Saccharomyces cerevisiae, FAS, also isolated from water kefir. The objective of this new study is to evaluate whether nurse bees fed with the yeast strain produce a greater amount of royal jelly in the queen cells. To conclude with, I would like to present you the members of the Laboratorio de Estudios Apícolas that participated in this study. Myself, María Agustina Rodriguez, my partner Matías Ledesma, Elian Thun, Francisco Reinaldi, and Leticia Fernandez, the director of the laboratory. Matías Ledesma is also working on honeybee nutrition, and he specifically studies the digestibility of the nutrients of the honeybee diet. And here I put a map of so you can see where are we located: Argentina in the south part of America, and Bahía Blanca in the south part of the country. Thank you very much for your attention. Here is the web page and the email of the laboratory and my contact information.